My name is Dr. Michael Ryan. I'm Coordinator of Science and Head of Vertebrate Paleontology here at the museum. And I want to welcome the public here as well as my colleagues from the Society of Vertebrate Paleontology who are hosting their annual meeting here in Cleveland at downtown at the Renaissance Hotel. I and the museum am co-hosting this with Case Western Reserve University, and the next person to meet up here is Dr. Darren Croft, who will actually be the other co-host. So thank you all for coming out and filling up our hall. I hope those of you who are on field trips today or were doing workshops as part of the conference had a great time. You did, right? Yeah, there we go. That's good. So the reason that you're here tonight is hear Dr. Neil Shubin talk about your inner fish. And that's part of a much bigger project, which Case Western Reserve University has started off to celebrate Charles Darwin, who was born in 1809. This next year, 2009, marks the 200th anniversary of his birth, and it also marks the 150th anniversary of the publication of his book, The Origin of the Species. So Case Western has taken it upon themselves in conjunction with people like our museum to bring in significant speakers who have advanced the field of evolutionary biology to give talks to the public to talk about their work and how that fits into the much bigger scheme. So this is an ongoing project, or a series of talks which will go through 2009. I should mention that the next talk is actually happening this Thursday evening on October 16th, and that is going to be Edward L. Larson, who's going to be talking about From Dayton to Dover, a history of evolution teaching legal controversy in America. And it'll be held over at the Case Western University campus. If you want more information, go to www.case.edu. I should also mention, because I'll forget this otherwise, that Neil will be actually signing copies of his latest book outside in the hallway here, in conjunction with our bookstore right after the talk. The talk will run for about 45 minutes, and then we'll take about 50 minutes of questions for Neil. I'll probably cut it off at that point, and then you'll generously let Neil get out to his book signing, and you can chat with him and say hello as you come up there. So without further ado, I'm going to introduce Dr. Darren Croft. He's a professor over at Case Western Reserve University in the Department of Anatomy. He's also a research associate here in my very own Department of Vertebrate Paleontology. So Darren, would you come up and do the honors?
It's really my distinct pleasure to welcome Neil Shubin here. I think you all have some idea of what Neil does, otherwise you probably wouldn't be here sitting in the audience. But Neil, for me at least, really personifies this field of evo-devo, which is a synthesis of evolutionary biology and development. And he is able to wear two hats, which most people are not able to do. He's able to go out and do field work and find fossils, and also be a gel jock, for lack of a better word, and have a real molecular developmental lab. And that's what you're going to hear about tonight. Neil's going to talk about what we can learn when we take fossils and put it together with the leading molecular techniques. What does that tell us about the evolution of humans and the evolution of vertebrates? Neil also has a distinct privilege of being associated with two great museums. He first got interested in paleontology, I think, when he was an undergraduate at Columbia University in New York, and so he spent a lot of time in the halls of the American Museum of Natural History. He then got his Ph.D. at Harvard. He did a stint at University of Pennsylvania as faculty, and now is in the Department of Organismal Biology and Anatomy at the University of Chicago, and he is also the provost at the Field Museum in Chicago. So two hats is probably fewer than what he actually wears at this point. So I'm glad he was able to schedule in a small amount of time to come here and talk to you about Tiktaalik and some of his other discoveries. So with that, please welcome Dr. Neil Shubin.
Thank you, Darren. Thanks to our hosts at the Cleveland Museum of Natural History, and thanks all of you for coming tonight. For my entire scientific career, I've been interested in the big steps, the big jumps in evolution. And when I went to graduate school, actually, I decided, one of the things I wanted to do for a thesis was to sort of understand one of these big steps in evolution. And after about a year or two, I realized, why not look at the origin of the first steps as one of the big steps in evolution? And it was largely by seeing this diagram in rough form by Len Radinsky in 1987. What I got interested in very early on was the fish-to-amphibian transition, or in this case, where you see a lobe-finned fish on top and an early limbed form on the bottom. And a diagram like this really captured my imagination, because what really is captured here is what we try to explain as morphologists, anatomists, as paleontologists, is to understand how major transformations, major changes can happen. And this diagram really captured my interest because, what you have is a fish-like thing on top. These creatures live in water. They have a very different way of making a living than the creatures on the bottom, which live mostly on land. And this is a giant jump in evolution. And I thought at the time that if we can understand new fossils, if we can discover new fossils, if we can discover something about developmental biology and genetics, we could begin to figure out how this seemingly impossible event happened. Now, in 1987, what Rudinsky showed here was essentially what we knew at the time. That is, the closest relatives of limbed animals, land-living forms, are these fishes here, the lobe-finned fish. And if you look at them, there are a number of differences between lobe-finned fish and amphibians, or limbed animals down here. And in fact, what we knew at the time was that lobe-finned fish were closely related to limbed animals, yet there were still major gaps in our knowledge in terms of some of the details about how the skeleton changed. So it became clear to me and a number of other people early on that one of the best ways that we could answer this question, solve this problem in understanding how the shift to land happened, is to discover new fossils. And to discover new fossils, what we really had to do is to try to find new places in the world that would contain creatures that would tell us something about this transition. And we did what paleontologists have done for over a century. That is, to try to identify new places to find fossils, we look for places that have sort of three things happening at once. That have places that have rocks of the right age to answer our question of interest. That is, in this case, it's pretty clear if you look at sort of the fossil record of lobe-finned fish and early limbed animals, that you want to look at rocks in the late Devonian, somewhere around 380 to 365 million years old. But also, it's not just rocks of the right age, it's rocks of the right type. Not every kind of rock produces fossils for a lot of reasons. Some rocks don't preserve fossils well, but also you may not have the right environments preserved in those rocks where the creatures may have lived when they were alive. So as a paleontologist, what you begin to learn is a catalog or a set of different kinds of rock or geological environments that are likely to contain fossils. So we look for places in the world that have rocks of the right age, rocks of the right type. And finally, we look for places in the world that have rocks exposed at the surface. I mean, it does us no good if our wonderful rocks of the right age and the right type are buried 5 miles underground. They have to be exposed to the surface. There's another variable too, and in my case, it was lack of money. What happened is, it's true. What happened is I started my first academic job here in Philadelphia, here in the southeastern corner of Pennsylvania. And I wanted a research program that I could do relatively on the cheap, with turnpike tolls and gas money, with the ability to just drive to a site. I wanted a local fossil site, if you will, that I could make some discoveries. And if you look at a sort of a geological map of Pennsylvania, stripping out all the uninteresting layers that are non-Devonian in age, what you see is in purple here is the extensive amount of Devonian that's preserved across the state of Pennsylvania. So it was very clear that sort of right about 3, maybe to 4 hours drive from my job in Philadelphia, we had the opportunity to look at some significant Devonian rock. And also, Pennsylvania not just had rocks of the right age and abundantly, but also had the right kind of rock to preserve fossils. That is, what you see here is a cartoon of what the eastern seaboard of the United States looked like in the Devonian. If you want to think about Pennsylvania and the Devonian, get Pittsburgh, Harrisburg, and Philadelphia out of your brain and think Amazon Delta. And what you had back then was highlands to the east, an inland sea to the west, much where we are today in Cleveland, in the Devonian. There's an inland sea out here, and a series of rivers and streams that drained from east to west, going all the way to estuarine systems. So it really became clear that this is a wonderful environment to find early limbed animals and their closest relatives, because we could sample ancient seas, ancient estuaries, and travel all the way up the ancient streams. There was another sort of variable that really helped the success of this early on, and that was when Ted Deschler joined my lab as a graduate student. Ted and I have worked together now for about 17 years since we got going in Pennsylvania, and much of what I'm going to talk about for the remainder of this entire talk has been done in collaboration with Ted and later in collaboration with Farish Jenkins. So when Ted joined, he wanted to do a thesis, and again, he was looking for safety over adventure, and so we started to look at Pennsylvania. And it became clear that Pennsylvania is wonderful for having rocks of the right age and rocks of the right type to find significant fossils. But it's also clear that Pennsylvania is not renowned for its exposures. It's not a desert. So our best exposures came when the Pennsylvania Department of Transportation decided to put in new roads. And this is one of those particular sites here. What Ted and I would typically do in the early 1990s is to see where the PennDOT was putting in new road work or a bend in highways, widening highways, and so forth. And we were very fortunate because they'd either just completed or were about to begin major expansion of roadways in central Pennsylvania. And this really helped us immensely. This is one spot here in Red Hill, Pennsylvania, which is about an hour north of State College. And what you see here is the Red Hill, right? So it's a red exposure. This was produced when PennDOT expanded a state highway in the early 1980s. And in so doing, they cut through a wonderful section of rock here. Here you see an individual for scale, some cars, and these are the different layers of Devonian rock. What you have preserved here is the life and life history of ancient streams. Each of these layers represents a different portion of the life history of an ancient Devonian stream. And as soon as we visited this site, pretty much in 1992, 1993, we started to find bones, particularly along these layers up here where this individual is walking. Some of the first things we found were, large sort of teeth the size of railroad spikes. Eventually, we started to find jaws of these creatures. This is Ted holding the front end of one of these jaws. Some of these jaws would be as long as your arm. The whole animal would be about 12 feet long. So we're pulling out these giant carnivorous fish out of the sides of these roadways in Pennsylvania. And we're also pulling out other sort of scaled fish. Here's a lobe-finned fish with its squashed head right here and portion of the side wall of the body. A number of kind of fish like this. And we're also beginning to pull out, and had done so over a number of years, limb bones, individual limb bones and portions of the skull of limbed animals. And what you see here is a humerus, an upper arm bone of one of these creatures that we discovered in the early 1980s. So what was coming out of Pennsylvania was actually a wonderful fauna and flora. And this is sort of the reconstruction that we did in the late 90s of what it looked like. This was done commissioned with National Geographic. And what you see here is some of the earliest forests and trees. You see a relatively small, shallow freshwater stream with that large carnivorous fish I showed you before with the big teeth. There were small armored forms of different types and a variety of different lobe-finned fish, including this large one, as well as some of the earliest limbed animals. But in terms of the limbed animals, actually, we were already finding pretty derived kinds of creatures. These were already creatures that had fully developed limbs. They were already creatures that actually had highly specialized limbs in many ways. We weren't finding what I'd like to think of as sort of creatures with transitional anatomies, sort of those creatures which would tell us about that transition I showed you about, in the second slide. So really what we were finding in Pennsylvania, in rocks of about 363 million years old, were lobe-finned fish and limbed animals, but really not very sort of forms with intermediate features, not transitional fossils, so to speak. And really what we wanted to get at here were creatures that really sort of bridge this gap. So to set to the stage for this, let me just go through some of the key features that we think about. The lobe-finned fish on top here that are most closely related to limbed animals, at least the ones we knew about in the mid-80s, had a conical head with eyes on either side. And one of the biggest shifts here that we see in the origin of limbed animals are changes in the architecture of the skull. You go from having this conical head to a head that's more or less flat with eyes on top. Furthermore, there are other proportional changes in the skull. You see this front end is actually relatively elongate relative to the back end. So there are all kinds of changes in the basic architecture of the skull that happen in the origin of limbed animals. Another big shift here, coincident with these changes, is the origin of a neck. Fish don't have a neck per se. What they have is a series of bones that attach the back of the skull to the shoulder. So that when a fish wants to reorient its head, it has to swim around. It can't move its head independently of the body. The origin of a neck actually happens when the head is freed from the shoulder and a series of specialized joints develop in the base of the vertebral column, base of the skull here, and the vertebral column, where the head can move independently of the body. So a neck is coincident with those changes in head shape I told you about earlier. And obviously one of the biggest shifts here is the origin of limbs, with fingers and toes and wrists and ankles. That is the transition from a fin to a limb, which involved not only the origin of new structures, fingers, toes, wrists, and ankles, but also the loss of other structures, which is the loss of fin webbing, which is so sort of abundant in fish fins. So I could spend the whole talk to you tonight just describing the differences between a lobe-finned fish like this and an early limbed animal. But you get the point. That is, essentially what we're trying to do is to find something that's intermediate in its morphologies. The state of affairs, more or less, at this time followed something like this. Now, this is a diagram showing relatedness, and what you have are limbed animals on top, so-called tetrapods, and then a series of fish groups that are successively more closely related to the limbed animals. We knew a lot, in fact, from our work in Pennsylvania and from colleagues' work elsewhere around the world. We know a lot about the stem of fish here, all the way from creatures known as rhizodontids up to these creatures here, which are known as elpistostegids. What Ted and I were after, really, were creatures that would bridge this gap, actually, the sister group of limbed animals. What would they look like? Now, if you deploy this diagram with first occurrences of fossils on the stratigraphic record, what you see is more or less there was a gap in our knowledge, and that the best place for us to look in terms of time for creatures that would bridge this gap would be at rocks about 375 million years old. That is, we were finding limbed animals in rocks about 365 to 363 million years old. If you look at the first occurrences of many of the creatures on that diagram I showed you before, that ranges in time from about 390 to 380 million years ago. These creatures all extend their ranges all the way up. But there was a gap in our knowledge, really, of creatures that would bridge this gap in a sense that would be closely related to limbed animals here at 375 million years old. So it became pretty clear that if we were to make a big dent in this problem, we would have to fill this question mark in time with new fossil sites. So Ted and I began really thinking about this just like we did with the cat skull. Let's look for rocks of the right age, let's look for rocks of the right type, and let's look for places in the world that have rocks that are exposed to the surface to try to find, to fill this question mark. We had all kinds of plans to work in the American West and a number of other places in South America, in Antarctica, and elsewhere. But everything changed for us one day in the winter of 1998 when we got into an argument. And to settle the argument, I pulled out an undergraduate geology textbook that was on my bookshelf, Evolution of the Earth by Dot and Batten, the second edition. This is the book itself. And to settle the argument, I was paging through the book, trying to find some ditty that would end the argument in my favor. And I found this diagram, which was to change the course of our research for the next 10 years. This diagram is so important and actually captures what we do in a nutshell that it's worth spending some time on for the non-paleontologists in the crowd. Basically, it's like figure 14.7 of Dot and Batten, and it says, Upper Devonian sedimentary facies. Now, when I saw this, I just stopped, because what I see is a map of North America. Here's the United States, here's Canada, Greenland. And superimposed on that are the Devonian age rocks and their likely environment that they were formed in or deposited in. So in the western part of North America, what you have is an ancient ocean or ancient seaway. And then you see three areas that were identified in this textbook as ancient delta systems. The first is here in the eastern part of North America. This is where Ted and I had been working all along, so we already knew that one. That was already fossiliferous. There was another area, so at the top here, East Greenland. Now, these were very well studied since the 20s and 30s and actually have produced some of the best examples of early limbed animals and some of their close relatives. These are creatures by the name of Ichthyostega and Acanthus Saiga, some wonderfully preserved and incredibly informative early limbed animals known since the 30s. But then there was another area extending about 1,500 kilometers east to west across the Canadian Arctic that I had never heard of and Ted had never heard of before. It just stopped us in our tracks because here we see an area that was mapped as late Devonian, extending across the Arctic, which if it's in the Arctic, it's likely to be exposed at the surface, almost like a desert. and virtually unknown to us and probably many of our colleagues. So that morning, we sort of stopped everything and went to the library and uncovered just a wonderful history. And that history actually began in the late 1800s. And it began with this sailing vessel, the Fromme. The Fromme, if you've ever studied Arctic exploration, polar exploration, this is one of the great ships of all time. Because it was designed by Cullen Archer in the late 1800s, and it was designed by him to carry Nansen furthest north. It's a special ship that can actually take great compression in the ice. And Nansen figured out that there's a current, if you will, that would carry a ship from Spitsbergen as close to the North Pole as anyone's ever been. And so this ship was designed to do that, to be passively carried to the north. This is in the late 1800s. So Nansen made a run for farthest north in this ship in the late 1800s. About 20 or 30 years later, Amundsen was to use this ship to travel to the South Pole and did his run for the South Pole. He was the first person to travel there. In the interim, in about 1902, a team of Norwegian explorers, headed by this individual, Otto Sverdrup, headed out in the Fromme, in this case to investigate what became the Canadian Arctic. And on the ship was this young man here in his late 20s, a pair shy. What these guys did from their expedition, this is from the account of their expedition, and around 1901, 1902, they traveled up to Ellesmere Island, Devon Island, and overwintered here for a period of time, exploring these areas, mapping them, and so forth. And on the boat, as I said, was Per Schei. And what Schei would do is he would get out when they parked the ship and do his geological explorations. And what Schei did was, in 1898 to 1902, in walking around the fjords up on southern Ellesmere Island and in local environments, he was getting bits and pieces of all kinds of different Devonian age fish. So here was an extensive area of Devonian that we were discovering that morning in 1998 that was visited by the Norwegians, and their young geologist, Per Schei, was uncovering a very significant fauna, albeit from bits and pieces of Devonian fish. This area was to see subsequent work by the Canadians in Operation Franklin and a geological mapping project in the late 50s. And then it was visited by this individual here, Ashton Embry, in the early 70s. Ashton Embry was to become the head of the Geological Survey of Canada's Arctic Mapping Division. But what Ashton did was to produce a beautiful paper in the early 70s that mapped the Devonian exposures in the Canadian Arctic just wonderfully. In fact, we would never have worked there if it wasn't for Ashton's work. So here's where we're talking about. Here's the North Pole. We're in the area of Nunavut. You could see it's outlined here. That's Flag Nunavut. And here you have Ellesmere Island. Zoom in on Ellesmere, and this is Ellesmere right here. So you're at a latitude of between 77 north and about 86 north. And what you see here is superimposed on this map are the Devonian exposures that Ashton Embry mapped. And here's the stratigraphic column for those exposures. And what got Ted and I going it got us so excited in 1998 was his description of this new formation, the Fromme Formation, right, named after that ship. He described the Fromme Formation as being identical to the Catskill Devonian rocks of Pennsylvania, only significantly older. So remember that question mark I was telling you about before in terms of time? What we are in here now is in the mid-Fronian stage of the Devonian. These are rocks that are about 375 million years old. The Fromme Formation fills that question mark. This all happened in one morning in 1998, and I was still at the University of Pennsylvania at the time. And Ted and I were so excited, because between finding the textbook and digging out these papers, we'd all of a sudden uncovered this wonderfully promising new area to work. So we went for lunch at a Chinese food restaurant. And I remember the fortune cookie I was to get at the end of the meal. It said, I opened it up, and I couldn't believe my eyes. It's still on my door. Soon you will be at the top of the world. So it was like, okay, my fate is totally sealed. We're going to the top of the world. Well, unfortunately for us, it's at the top of the world, but it's here. Okay. It's mapped as obviously as delta sediments, but it's very similar to the Catskill we're working here. But the toolkit we used in the Catskill in Pennsylvania obviously would not work well for us up here in the Canadian Arctic. You know, you're dealing with a latitude that's about 78 to 80 north. It's daylight 24 hours a day in the summer. There are polar bears up there. There are logistic problems about getting there and coming home and getting fossils out. So there's a whole new toolkit we had to develop or apply to work up there. To give you a sense of the challenges, you know, there's a town about 200 miles from our fossil sites. And this is a picture of that town. It has about 170 people in it. This is a picture of that town in spring. Okay, it's Grise Fjord, Canada. I mean, that's civilization 200 miles away. So it became clear that Ted and I had a great idea, but getting there and working there is a whole other issue. Now, I had worked in Greenland on the Triassic rocks for a number of years, and I had the great privilege of working with Farish Jenkins, my former advisor from graduate school, during those Greenland years. And what Farish and the MCZ field crew, the Museum of Comparative Zoology field crew, did was they developed a wonderful toolkit for working in the Arctic, a lot of know-how to get there. So it became pretty clear that this was going to be a three-way collaboration relying on Farish's expertise and our enthusiasm and ideas and all of our money, or our ability to try to raise money to get there. So the team was set. It was Farish, Ted, and myself. And we visited in the early 90s for the first time. The trick in working there is obviously logistic. We get around with these planes here. This is a twin Otter. It has a stall speed of about 55 miles an hour, so it can take off on land on tundra in very short distances. And our sites are so remote that a twin Otter can't really land in some of our sites, and so we have to ferry with helicopters into our field sites. So it's this continual shuffle between landing with the twin Otter and getting the helicopter into the sites. What all this means is, and this is really important for the science that we do, is that our weights are very limited. We can only bring a small amount of stuff, and unfortunately, because fossils are so heavy, we have to make real decisions in the field about what comes home with us. Well, this is what camp more or less looks like before we set it up. We take a small number of people and a small amount of stuff. Food gets sealed up in these tubs, which holds most of our supplies and actually keeps things reasonably smell-free from the polar bears that are out on the ice. And we involve local Inuit as much as we can. In a few years, we've had Inuit youth from that small village I showed you before joining us. So it's been a great experience for everybody. But the key thing is obviously that we have to be very careful with weights. And the way that impacts the science is, unfortunately, when we're working in the field site, it oftentimes feels like a salvage operation. That we're trying to say, do we want to take this home or not? And it's very frustrating at times to deal in that circumstance. So the fortune cookie we had in 1998, and the first year we visited there was in 1999. And we were all excited, because here we had this great idea. We were fortunate enough to be able to raise money. And we decided, well, we'll begin in the western part of the Arctic in Melville Island here. So Ellesmere is here. We began on Melville. And this is what camps looks like. We have a cook tent. We have a series of smaller tents. We each have our own individual one. Usually camp close to a snow field with running water, because you can drink the water right out of the streams. And you get a sense of what we do from here. Here you could see the Devonian exposures. They're pretty low-lying, at least in the western part of the Arctic. And we'd walk over these things day in and day out looking for bones that are weathering out on the surface. The problem with 1999 is we really didn't find much. We went out there to the western Arctic, and we had bad weather pretty much for 2.5 solid weeks. Furthermore, we realized that we plunked down in the wrong part of the Arctic. We plunked down in the western part of the Arctic where there was an ancient seaway. So where we were finding fossils, we were actually in very marine-like sediments, oceanic sediments, kind of much like you would find just south of Cleveland. So it became very clear that if we're going to make any headway, we have to go upstream, if you will, up the ancient streams, which meant, in this case, going east. So in 2000, we went east. And that's when we first started to get to southern Ellesmere Island. This is what southern Ellesmere Island looks like. There's camp that year. This is Goose Fjord. One thing that's relevant about Goose Fjord here is this is where that ship, the Fromme, overwintered one year. And this is the exact same spot Parish did some of his initial investigations, finding little bits and pieces of bone. Boom, as soon as we moved east, we got into the river kind of environments and lake environments that we figured would be so productive. And as soon as we hit those, we started to find lobe-finned fish. So it was really a big move for us to go to southern Ellesmere and further east. Now the problem with this is this all looks like great rock to work on, but the reality is maneuvering in the Arctic is extremely hard. And this slide sort of captures it in a nutshell, is that what looks like great exposure are usually these awful talus scree slopes. The kind of scree that's kind of loose and unconsolidated, where you take one step and the whole thing sort of slides down. So getting about there is actually pretty tough, because when you don't have this sort of nasty scree, you often have pretty horrible mud. So it's this, the walking is never entirely easy up there. The other thing that's never entirely easy is the camping itself. And this is a slide I took in 2000. And what you see here is our North Face Himalaya Hilton. It's more like a Himalaya Motel 6, but that doesn't do justice to Motel 6, because what happened is we had high winds and it ripped the fly off the tent. This is my tent after those storms, and we typically put big wind walls up. But weather can be a problem there. We will typically lose about 1 day out of every 4. And so that's to wind, snow, fog, or what have you. There are polar bear in the area, so we carry weapons. And setting up these wind walls actually turns out to be quite essential, because without these wind walls, these tents would have blown away. So what we do is we walk around on the rocks when the weather's good, and this is just a picture taken from last year. And what we look for is what paleontologists everywhere look for, is bone weathering out on the surface. And what I hope you can see in this slide is, you see everything that's a little shining here? This light was perfect for this kind of slide, because what you see, what's shining here, everything that's glowing is bone. Because if you zoom in on it, what you could see is here's the native rock, and here's bone weathering out of the surface. So what we're looking for is bone that's weathering out of the surface, and then following it to a layer that produces whole skeletons, or hopefully whole skeletons. Well, the big event in this whole fieldwork happened at the hands of our youngest field crew member. Jason Downs, who at the time in 2000 was an undergraduate at the University of Pennsylvania, real eager to learn paleontology, so he joined our crew. And this is the site Jason was to make the huge discovery 4 hours before Jason was to do it. What was to happen here is about 3 or 4 hours after Ted took this slide, Ted just got up and sort of took a picture, just for our vista. And little did you know, he was recording a little bit of history, and that 3 hours later, Jason was to walk here and make a huge discovery. But we didn't know that at the time. What happened was Jason was late back to camp that night. Usually we ask everybody to be back in camp by around 6 o'clock. It was around 7-ish, and Jason was still late. You know, where's Jason? Since he was the youngest field crew member and he was separated from the rest of the crew, we were actually getting pretty worried. And it wasn't until, you could hear his footsteps, and you hear him open up the zipper of the tent, comes inside. Then you could see his eyes and his pockets. His eyes were glowing, and his pockets were filled with fossil bone. Turns out what happened is, Jason came back and he starts pulling out fossil bone after fossil bone on the table in front of us. What happened was, about 3 hours after Ted took this slide, he was to walk here in this little green band. And that green band was mostly green because it was covered with thousands upon thousands of fish bones. Jason had digital images that he took that day of this site, and they were shaking so much because he was shaking so much. We decided, well, let's skip dinner. We grabbed candy bars, and this is us that night visiting Jason's site. And what essentially we did was we just do what paleontologists do all the time, which is crawl up and down this hill to try to find the layer that Jason's bones were coming from. That does not sound like rocket science, but that took us weeks to do. And the reason why it took us so long to do was these nasty cracks here. What you have in the Arctic is a situation where it's really cold in the winter, and in the summer, it's less really cold. So you have this freeze-thaw effect where it goes really cold, less really cold, really cold, less really cold. And in the process, the rocks are expanding and contracting in situ. You do that over a number of years, and what happens is you end up with an upper layer of rock that's like churned. And so what we would do is, every time we dug for Jason's lair, we would hit these churned layers where we weren't in any real bedrock. And we had to really clean the whole area off over a long period of time to find Jason's lair. But find it we did. It took another field season to do that. And this is Ted here, Marcus Davis, Corwin Sullivan, who opened up this layer. And you could see there's the layer, Jason's lair, being exposed. And as soon as that layer was exposed, it became clear that those individual fish fragments were produced by skeleton after skeleton of fossil fish, many of them lobe-finned fish, buried one on top of the other. Some of them relatively complete, some of them partial, but all of them very good quality bones. So then the objective became, let's remove as much of this rock as we can. So this is the site here that Jason was to walk on. This is what it looked like in 2006, where we opened a big notch to expose the layer that Jason found. Now the first really big discovery happened at the hands of this gentleman here, which is shown to his back is shown to you. This is Steve Gatesy from Brown University. Here's Farish's back right here. One day, Steve's picking up rocks, and he says, guys, what's this? And I don't know if you can see it very well, but here you could see, this is the rock here, but there's this little portion here. And if you look at it really carefully, there's one bone going like this, there's another bone going like this, and there's another bone here. Looking carefully at it, what it became very clear that this thing was a snout of a fish sticking out at us. And not just any fish, a flat-headed one. So you remember I showed you before, conical head to flathead? Here we had a flathead sticking straight out at us, which meant not only did we find, we think, what we were looking for, but also that if we had any luck, that the rest of the skeleton, or a good chunk of it, would be preserved in the remainder of the cliff. And so what Steve proceeded to do, and this is shown a few weeks later, maybe about 10 days later, is to etch this thing out as a giant pedestal, sort of roughing it out to hopefully have some skeleton inside. And he was aided by the fact that he'd occasionally find a little scale of this creature here on one side. So it seemed clear that we had, with any luck, would have a good chunk of the skeleton. Well, as Steve was doing that, we were discovering other flat-headed fish like this one. I discovered one at the bottom of the hill. Farish here discovered another one right next to Steve's specimen. So before we knew it, we had 1, 2, 3 of these creatures coming right out of the hill. The trick became to get them home because at the time we were using plaster, and plaster in the Arctic is a very bad thing because it never dries. And it's made even worse by the fact that these things had to come home on the bottom of a helicopter. But come home they did, and it was here at home where the preparators, Fred Mullison and Bob Masick, got to work on these things. It took Fred for this specimen, this is Steve's specimen, it took Fred about a month to get to this point, picking away with a needle and a pin vise, removing the grains of rock. And all of a sudden we start to see, well, here's where Steve's snout was, Steve's fossil snout. Here's the top of a head with 2 eyes on top. A few months later, that's what it looked like. Here's the head, there's the orbits. It looked like we were getting a shoulder girdle out. Now you remember what was going on at this time period, that these fossils were being prepared. While these things were being prepared, there was a trial going on in Dover, Pennsylvania. And that trial, as many of you may know, was to support the teaching of intelligent design in the schools. And one of the arguments you'd occasionally hear is that there are no transitional forms in the fossil record. There are no fossils that tell us about transitions. Well, this thing's being exposed like this while that trial is going on. And during that time, this was going on in my head. Here's a fish, here's an amphibian, here's the new fish. And it was truly remarkable. This is the creature that came out. In fact, this is the exact same specimen I showed you that Steve was working on. In fact, the snout he exposed is right here. If I was to hold this particular specimen in front of you, it's a creature this one is about 4 feet long. You see it has scales on its back and fins with fin webbing, so fishy kind of stuff. But as I showed before, it has a flat head with eyes on top. It has a neck where the head is separate from the shoulder. And when you remove this fin webbing, what do you see? You see bones that correspond to our upper arm, forearm, even portions of things that became our wrists. So it's truly a mix of fish and limbed animal characteristics. So this is where it fit in the scheme. So you have things like this creature here, the Eusthenopteron, from the first appearances are a little over 380 million years ago. You have limbed animals up here at around 365-ish so million years ago. And this thing in the middle, which like a lobe-finned fish, it has fins with fin webbing, scales on its back, and primitive jaws, among a list of other features. And like a land-living animal, or limbed animal, it has a neck, portions of a wrist, a flat head, and other features, which I'll show you in a second. Now, as discoverers of this creature, Ted, Farish, and I were privileged to give it a name, as discoverers of fossil taxa often are. And what we wanted in this case was the name to reflect its Inuit heritage, its provenance from Nunavut. I mean, we had worked there at the pleasure of the Inuit people. They had helped us. We had worked with the youth from the town. So we wanted the name to mean something in their tongue, Inuktitut. So we talked to the Inuit Council of Elders. And we worked with them and discussed with them to really come up with a name that does two things. One, a name that means something to them and to us. And two, a name that we could pronounce. And the name of the committee did not lend me a whole lot of confidence that we would come up with a name we could easily pronounce. Anyway, so I'm talking to one of these, that was my job to come up with a name, talk to the Inuit and so forth. So I was talking to one of these folks here, and we had a really hard time communicating what this was. Because when I first talked to him, I said, well, you know, we have a fish that we want to give a Inuktitut name to. He said, no problem there, we can help you with that. I said, well, we find this fish in sandstones and rocks. He's like, no, hunters find fish in streams, and like not in rocks. So then I realized, well, we have to explain what a fossil is, actually. So it took a while to really sort of convey what we did. And actually, it was really hard. And eventually he said, look, just tell me what it is and where does it live. I said, well, it's a large freshwater fish. He says, oh, why didn't you just say so? You got yourself a Tiktaalik. I said, a Tiktaalik? What's that? He says, a large freshwater fish in our language. I kid you not. And there was another name he came up with that means the same thing. It was like a synonym, which we couldn't pronounce. So on all those virtues, Tiktaalik stuck. Now, Tiktaalik, one of the advantages of finding Tiktaalik is, as I told you, in 2004, when we discovered them, we had at least 3 partial skeletons that came out. Since then, we've discovered portions of about 30 individuals. So it's well known from, I can't say it's abundant, but it's well known from a number of different individuals. Several of those are partial skeletons, well preserved. This is one of them. This is, what we've now seen is that Tiktaalik ranges in size from the smallest specimens, which are just under about 4 feet long, to the biggest specimens, which are just over 9 feet long. And this is one of the 9-footers here. And you're seeing the skull in ventral or underside view. And here you see the lower jaws. You're looking at this view here at the lower jaws. And we realized that we can now take them apart. Being able to work with great preparators like Bob and Fred, and having relatively abundant material means that we can take these things apart and look at the bones in some detail. So that's what we did. And in doing that, we discovered here's a shoulder, here's a humerus, an upper arm bone, there's a radius, and there's an ulna. We were able to take apart several different fins and look at them in some detail. In doing that, and this took a considerable amount of time, each of these bones was removed. So here's the humerus of Tiktaalik, there's the radius of Tiktaalik, ulna, intermedium, ulnari, and a series of radials. This is what we knew in 2006. We now know that there's a whole array of radials that goes out here and another row out here. So it's a fully formed fin. But not only can we learn the pattern of these bones, we can actually look at the joint surfaces of these different bones of the fin. So here's the shoulder. Here's the shoulder socket, the so-called glenoid. Here's the head of the humerus and how it would fit in. So this is the shoulder of Tiktaalik, seen with both portions of the joint. Here's the elbow of Tiktaalik, seen in both portions of the joint. And here are the 2 portions of the wrist, the so-called proximal carpus and the more distal carpus. In looking at this, we can begin to see this animal as an animal that's able to move around. And what you see here are the shoulders of Tiktaalik, as well as other animals it's closely related to. So here are some fish further down the tree. This is a creature called Sauropterus. Here's Eusthenopteron. I've showed you examples of this before. there's Tiktaalik, and here's an early limbed animal, a creature from Greenland known as Acanthus Saiga. When you look at this series, so think about marching up the evolutionary tree, if you will, as you look from Sauropterus to Eusthenopteron to Tiktaalik to Acanthus Saiga, what you see is the shoulder socket goes from facing backwards. You can see it here, and the reason why you can't see it fully is because it's facing backwards, to facing laterally or towards you in Acanthus Saiga. Okay? So what that means is the fin sticks backwards in Sauropterus, backwards in Eusthenopteron, yet in early limbed animals, it sticks out sideways, much like this. So the series is from facing backwards to facing to the side. What does Tiktaalik have? It has a joint that's facing sort of intermediate between these. It's facing not backwards entirely, not completely laterally towards you, but sort of halfway between both. There's another important way Tiktaalik is very sort of intermediate in its form here, and that is the kind of bone that makes up this shoulder girdle of Acanthus Saiga is fully formed in cartilage first, it only forms in cartilage first. Whereas if you look at these fish, Sauropterus and Eusthenopteron up here, what you find there are two portions of the shoulder. There's a small portion that forms in cartilage first, and another portion that forms from just ossification in just connective tissue. Tiktaalik is intermediate in this regard, and I'm not showing you the right view for this, but it has a very well-formed, if you look at the internal surface, you'd see it even better, well-formed cartilage portion of the shoulder, and a relatively smaller membranous or portion of the girdle that does not form in cartilage. If we put the humerus, the upper arm bone, on these things, what you see is sort of a rotation of the humerus in its sort of resting position from Sauropterus and Eusthenopteron, where it faces backwards, to facing towards you in Acanthus Saiga. So here you see the elbow joint looking at you, to Tiktaalik, where it's oriented sort of halfway between facing backwards and facing towards you. So if Acanthus Saiga is facing out at you like this, and Eusthenoptera and the fish are facing backwards, Tiktaalik is sort of 3/4. If we look at the elbow joints, actually something very interesting is happening to the elbow joint here. So we can see the elbow joint in Tiktaalik. Let me just click back a bit. You can see the elbow joint of Tiktaalik right here. And what's remarkable about those is the way that these facets, the portion of the joint, rotates to the belly side. What does that mean? What that means is when you put the elbow together, its natural position is slightly flexed, okay, sort of slightly bent. Unlike Eusthenopteron and Sauropterus, where the fin or the elbow is actually relatively flat, it's actually more flexed like an early limbed animal. Okay, so you already have an elbow that is as capable of a degree of flexion not present in these fish. And then when we add the digits in Acanthus Saiga and these elements that sit in the farthest reaches of the fin in Tiktaalik, Eusthenopteron, and Sauropterus, what we find here in Sauropterus is that the joints in the sort of the distal, or the portions of the skeleton that are furthest away from the shoulder, that these are capable of extension. What you have is, in Tiktaalik, is an animal that's able to do a form of a push-up to prop its body up. That is, it can flex the elbow and extend at the wrist, much like this. So when you do a push-up, what happens? The palm is flush against the ground, your wrist is bent, and your elbow is bent to enable you to do that activity. That's exactly what you see in incipient form in Tiktaalik from the analysis of those joints. And we would have never known that if it wasn't for the work of our excellent preparators. So Tiktaalik is already along the way to having a very limb-like design. But Tiktaalik is not a walker. Okay? Tiktaalik couldn't walk around like something like Acanthus Saiga or Ichthys Saiga. And the reason why I say that is if you look at the shoulder, it's not capable of extensive protraction and retraction. It's a very limited shoulder joint. Only a moderate degree of flexion and extension is possible. So it seems like Tiktaalik's able to do a push-up and prop itself up with its appendages, not fully walk like later limbed animals. The other thing we're able to do is, with the number of specimens we have, is lay on the fin webbing. And this is where things get very interesting. When you think about a fin of a fish, it's composed of several different kinds of bone. There's the bones I've been talking about, such as the upper arm bone, forearm bones, and wrist bones. Those are bones formed in cartilage. Then there's the fin webbing. The major change we see from fish to limbed animal is a loss of the fin webbing. If you look at a fish fin, what you see is it's mostly composed of webbing. Okay, like look at a trout or or a salmon, what have you. And indeed, even if you look at these lobe-finned fish here, you find an extensive amount of webbing. It's completely lost by the time you get to Acanthus Saiga. And the question is, what does Tiktaalik have? Tiktaalik's reduced it. Tiktaalik's reduced it in a very particular way. It's gotten stubbier, but also Tiktaalik has lost the fin webbing that would cover the elbow and wrist joints, meaning that what it has is the ability to flex and bend. Yet it's kept the ability to swim around and form a broad plane that a fin with fin webbing would do. I just want to step back just to show you how we know that, because this just testifies to the fossils themselves. So here's the specimen of Tiktaalik I showed you earlier that's shown in view from the top. This is the specimen that Steve Gatesy found. And here is the fin. If you zoom in on that fin, what you can see is here's the fin webbing. So what we were able to do with this specimen and others is look at the relationship of that fin webbing to the bones on the inside. And it's through that we were able to see that the functional joints that allow that push-up motion aren't covered by fin webbing. So what we're seeing at Tiktaalik is the way that the transformation from lobe-finned fish to limbed animal happened. And we're seeing it most clearly in the appendages up until 2006. Then recently what we've been working on is the skull, and what will be Jason Downs will talk about tomorrow in the Society of Vertebrate Paleontology meetings, and which will come out in Nature this week, is an analysis of the brain case and underside of the skull of Tiktaalik. And just to summarize some of that for you here, what you see here is Eusthenopteron, a lobe-finned fish on the bottom, and here's Acanthus Saiga, a limbed animal on top. And what you see is the major changes, some of the major changes that happen here are in proportions. So you're finding the snout expanding as you go from fish to limbed animal. And you're finding the back end of the skull reducing. In particular, one of these bones reduces dramatically. It goes from being a large, sort of boomerang-shaped bone to a small little spicule here, which is known as the stapes in Acanthus Saiga. It turns out Tiktaalik is intermediate in both these conditions, that it has a skull which has sort of the proportions of an early limbed animal with an elongate snout, but still retains many of the proportions in a fish. Also, the hyomandibular, this bone shown in blue, is almost intermediate in size between Eusthenopteron on the bottom and Acanthus Saiga on the top. We also see in this skull the complete loss of the opercular series. These are bones that would serve as flaps to be used in gill breathing. The loss of the opercular series is associated with the origin of the neck. That is, by losing those bones, as well as a number of other ones, we now have a creature that is able to move its head independently of the body. When you think of these features, what you think about is a coordinated set of changes happening that impact all the functions of the animal. That is, when you look at the fin, you find a fin that is able to support the animal by propping itself up. Associated with that is the origin of a neck. Well, clearly a neck becomes useful in creatures that are supporting themselves by their appendages. Because fish can, as I said earlier, can swim around and reorient their eyes. According to how they swim, they can reorient their mouth and eyes appropriately. Whereas a creature that's doing a push-up on the ground, the only way it can look around is with a neck. And it's interesting that as soon as we find an appendage that can support the body and prop an animal off the ground, we find the origin of a mobile neck. So it's useful to think of Tiktaalik as an animal that is living in both in the shallows and on the benthos, that it's living on the bottom of the water and in the shallows. It has both gills and lungs. It has scales in its back and fins with fin webbing. Yet, when we remove those things, we find a head that has proportions that approach those of an early limbed animal. It has a neck, and it has a very complex appendage with an upper arm, forearm, even parts of a wrist. There are other portions of this that I haven't talked about, and that what you see in Tiktaalik is an extensive set of ribs that overlap with one another, which really actually fit together, tongue and groove, almost like they're supporting the trunk under a gravitational load. Since the discovery of Tiktaalik in 2004, we've actually discovered more material, and we now have no pieces of the hind fin that weren't described with the original announcement, and so we have a pelvis and portions of the hind fin. We're lacking a femur at the current time, however. So we did our our reconstruction of Tiktaalik. But other people did a reconstruction of Tiktaalik as well. When Tiktaalik was announced in 2006, it came with an enormous amount of news coverage. It was the lead story in the New York Times, you know, had 2 appearances on the Colbert Report. I mean, it was just one of these fossils that was everywhere. I couldn't forget about Tiktaalik for a period of time. But one of the beautiful things about that was, remember the social context where Tiktaalik was discovered, which was the people saying that there are no transformational evidence of transformations in the fossil record. And here, we designed an expedition to find a transformational fossil, and bingo, we found it. And not only that, it was loaded with surprises. It answered questions, but it enabled us to pose whole new ones. Well, when that story came out, one of the most rewarding pieces of that was our ability to work with schools, and to work with everything from high schools to junior high schools. And you know, Miss Philbin's 3rd grade class in Manchester, Vermont did a paper mache model of Tiktaalik, not far off from one of the 9-footers, mind you, but a wonderful way to bring evolution into the schools through discovery, which was for us, all of us involved in the project, very powerful. So finally, to close, I just want to make a sort of personal comment, and that is, you know, all too often I deal with people outside of the field of vertebrate paleontology who think somehow that the great discoveries have already happened, that they happened long ago. And you know, it's so surprising to them when I tell them that some of the greatest discoveries in the history of paleontology happened in the last 15 or 20 years. Think about what we now know about the origin and evolution of mammals just from discoveries in the last, say, 15 years. Think about what we now know about the origin and evolution of early vertebrates, of list amphibians, of dinosaurs, of birds. I mean, these are things we could have only dreamed about when I was in graduate school. So it's really remarkable that we live in an age of discovery in paleontology, and seeing, you know, the topics that are going to be discussed at the Society for Vertebrate Paleontology meetings over the next few days, it leads me to think that actually just come back in 20 years, that we are living in an age of discovery, that we're only beginning to see the fruits of it. And so we're really going to understand so much about the history of life from the work that paleontologists do. Finally, I also want to close with an image, and it's about the Arctic. This is a picture I took in 2002 in Oxy Bay in Ellesmere Island. You'll notice this little hill here. But anyway, I took this picture. I don't even remember why I took the picture, but there are footprints. These are my footprints. We visited the site again 8 years later, and you'll notice see this hill here? This is this hill. And by some dumb luck, I took a picture of this site, and you'll notice there are my footprints. 8 years later. Working in the Arctic, you cannot imagine just how special a place it is. It's wonderful exploration because you feel like you're walking the footsteps of some of the great explorers. But not only that, you feel you're in an environment that is extraordinarily fragile. And to me, this image here of footprints captured in the mud really sort of brings home the point that the Arctic is a wonderfully special and unbelievably fragile place that I can attest, because I've been there this last year, is increasingly threatened by human activities. So just to close with that, because I also think the Arctic is a place where many people can make many discoveries ranging from Mesozoic all the way to Paleozoic. So I look forward to following that with great interest. Thank you very much.
Very briefly, did you consider naming your creature Dover?
Yeah, really. Yeah, exactly. It was, I mean, it's a remarkable experience. I just used that as a template for something else. I remember the experience I felt in reading about the Dover trial, and there was the cast of the Tiktaalik skull sitting on my desk. It's like, oh God, I wish I could tell somebody about this.
Thank you, Neil. That was a beautiful talk. So you were very careful when you were describing the hand part of, I shouldn't even call it the hand, the distal part of the limb in the Tiktaalik. You were careful not to use the word digits. And even though they're articulating joints, do you think they have something like a hand or the beginnings of a hand? Because they have the wrist bones, but would you call the pile of bones distal to that actually digits in the sense we understand it?
Yeah, what they have is they have transverse joints. They could do this. They had a row of what you'd call phalanges, right? I mean, what I didn't even show is because what we've known in the subsequent years is that there's each of those rays has multiple bones and several joints. They have the ability to have a palm that lies flush against the ground. At least 4 of those bones are homologous or comparable in an evolutionary sense to the to bones in limbed animals. So I would say there's a good portion of the distal appendage which is homologous. Are they digits? Now, when we think of digits, we think of as a particular kind of developmental process that's involved there. And one of the things we've discovered, my lab and a number of other labs, is the genetic address of that portion of the appendage is virtually identical to that which makes digits in chickens. And we see it in basal or ray-finned fish, which are close to the transition. Subsequently, some people have found it in lungfish and in other fish as well. So it really appears to be the genetic machinery that makes digits is actually present before we see them in the fossil record. Yet what we see in Tiktaalik is something that I think you can compare very directly functionally to digits. You can compare them spatially to digits, where they sit in the appendage. You just can't compare them in terms of their shape. They're not elongate. They're still very stubby, which is one of the reasons why I love doing field work, because I really believe that if I go, as we go to younger rock, which is exposed up in the Ellesmere Island exposures, and we find a limbed animal up there, or a proto-limbed animal, we'll find something that will bridge that gap between the appendage of Tiktaalik and Acanthus Saiga.
I was looking at the movement of the opercular bones, hyomandibular area, and it made me wonder about what you were able to see pertaining to movement of gill arch members.
Yeah, so you have the full set of branchial elements. I didn't show them, but what Jason might show tomorrow is the full set of branchial elements of Tiktaalik, all of them that are preserved at least in bone. There may be others that are preserved in cartilage, which we won't have. But what you see is this median element. So what you have in Tiktaalik is it's not unlike other lobe-finned fish, actually, except the fact it's much more robust. That is, what you have is a median set of elements that sit in the midline, and then these gill arch bones that stick away from them. These are the branchioles. The central element is massive and has a huge crest on it for muscles, implying that what you're having is an emphasis in Tiktaalik on the amount of muscle power that's devoted to pumping the gills, which makes total sense. Because when you think of how many ray-finned fish breathe, what do they have? They have 2 pumps. They have an opercular pump that works in coincidence with a pump in the mouth area, the throat, the pharyngeal pump. Okay? What you have in Tiktaalik is loss of the operculum and reduction probably of the opercular pump. So now you have an animal that's relying mostly on mouth breathing, mouth pumping, I should say, of water, likely air as well. And a coincident with that are changes in the shape of the head, right? It's a wider head, but also the amount of muscle crest and clearly the amount of muscle mass devoted to pumping of the pharyngeal area, particularly the branchial skeleton.
I'm not a paleontologist, but I've thought a lot about the anti-evolution argument of a lack of a missing link in the fossil record in this context and in a variety of other contexts. And to me, this seems like a trick question, and it seems to be fundamentally rooted in the difference between, if you go back to basic statistics, between continuous variables and discrete variables. And if you think about a lineage moving through time, it seems that it would be undergoing a slow change throughout its history, and therefore be similar to, at least conceptually, to a continuous variable. So if an anti-evolutionist wanted to make an argument that there's no missing link, as soon as a new a new missing link is discovered, the anti-evolutionist could easily just pick 2 more points somewhere on the lineage, and there would always be a missing spot. Now, my question to you is, this is great that you found this. Now, are the anti-evolution folks going to say, well, that's great, but there's no link between these 2 lineages, and it's just going to keep going on and on? And if you think that's the case, have people in your position consider this and consider this possibility and thought about this as a solution to their argument?
Well, one of the things, when Tiktaalik was described, I had about 15 emails in my email box saying, congratulations, you discovered 2 new gaps in the fossil record, right? And which is, true. And And what happens, I approach this as somebody who's exploring, right, who's trying to discover new stuff. And I approach this as an issue. Whenever I make a new discovery, whether it's Tiktaalik or something else, or somebody else makes a new discovery that I learn about, what it does is it answers old questions, but gives me the power to ask new, more refined ones. And that's exactly what Tiktaalik has enabled us to do, as well as every other creature that's associated with the, fish tetrapod transition, whether it's Acanthus Saiga, Ventus Saiga, all these stegas and ichthyos and so forth. Each one of those has, in its time, answered questions as well as given us the ability to ask powerful new questions. So the kinds of questions I'm asking now about fins, about breathing, are questions I wouldn't have been asking the same way 2 years ago. I've answered old ones and I'm posing new ones, and that's the process of scientific growth. It's never a, people sometimes think that, well, you've stopped answering the questions. Why are you even looking at the Devonian now? And I say, you couldn't have it more wrong. The reason why I'm looking at the Devonian, even after finding Tiktaalik, especially after finding Tiktaalik, is now I know what kinds of questions to ask with greater precision that I know I'll be able to answer, because I now have a sense of what the tools are in the Arctic to answer those questions are, the rocks and so forth. So I look at it in a slightly different way. I look at it in how can I make new discoveries and how can I pose new questions.
You said that the amount of weight that you took with you during your trip to and from the Arctic was very crucial, because only a limited amount was allowed due to the limitations on transport. How much weight did you initially carry with you into your trip, and how much did you end up returning with?
It's about 1,000 pounds each way. So we judge it by the number of helicopter runs, and so it's about 1,000 pounds of stuff, and that includes food. And so what happens during an expedition is you bring in 1,000 pounds of stuff, and the food weight goes down, and the fossil weight goes up. And that we hope that those equal out. They don't always, so we have to leave fossils up there from time to time. So that's what the calculation usually is. We also try to build in some sort of error factor there, safety factor, which has gotten increasingly hard. This past season was very difficult for us because the rise in oil and gas prices coincided with a drop in the dollar versus the U.S. dollar versus the Canadian dollar. So we were hit in multiple ways, and so we actually had to reduce the size of the crew this year and told everybody to lose weight, so. Myself particularly.
Why would the skull change such that the orientation of the eyes would be on the top of the skull as opposed to the sides?
Well, there's a couple reasons for that. I mean, if you think about what Tiktaalik's doing, it's an animal that's supporting itself with its appendages, right? And it could be using that in two different ways. It could be living in the water bottom, looking up. So either way, it's looking up, right? It can either be at the bottom of the water column, looking up at the fish that are swimming by, right, as a search and wait sort of a predator, or it could be like in a crocodile niche with its eyes out of the water, feeding on the large invertebrates that were around at the time. Flathead with eyes on top is actually in a relatively flat animal. I should say it's a relatively flat animal. Useful in a creature that's living in that interface between water and land. In the shallows, where the difference between the water bottom and the water surface is not that great. A lot of this is, we believe, related to the shallow water environments that Tiktaalik is adapted to.
I have two questions. The first is, why would the arm limb develop first as opposed to both limbs at the same time, or the pelvic limb first? And the second is, what are the developments in the brain from lobed fishes to the limb?
Okay, I can answer, the second one I can only answer indirectly. And that is, what you find is a change in the sensory apparatus as you go from fish that lives in water to animal that lives on land. You think about it, sensory perception in water is very different from sensory perception on land. So if you look at a picture of the skull of Acanthus Saiga, which is the early limbed animal, and truly aquatic creatures, what you find is orbits are all of a sudden really huge. So it seems to be there's an emphasis on a particular kind of vision. And so the sensory kinds of changes that happen appear to coincide with water to land and the different kinds of needs that these animals have. In terms of the appendage, what was the question about the appendage is why the orientation?
Why the forelimb as opposed to?
Well, I got to say, we don't know for sure. I got to tell you, the hind fin of Tiktaalik could have some surprises in that regard. That being said, the current theory is that that these animals, pull themselves around with their front appendages. They still have a tail. Okay, so so we still have a very aquatic body form with a tail that could be used for aquatic propulsion. So the idea is the animals that the forefins evolved to support the animal for multiple reasons. It could be to prop the animal off the substrate, the bottom substrate, so it can breathe with its lungs, to enable the animal to lunge forward in conjunction with the tail, and so forth. But that's a case where I think good new fossils will tell us a lot of new insights.
I think Tiktaalik's a great example of targeting an unsampled interval in time, kind of what you intended to do. What's the rest of the fauna look like, and have any of those also filled in gaps in the lobe-finned fishes or other groups? So
there are a lot of placoderms. There are a number of armored fishes in there. There are porolipiforms, other large lobe-finned fish. There's a really large lobe-finned fish in there, a so-called Tristicopterid. We have about 12 different species of fish that are coming from there. No invertebrates yet. The smallest fish that we find in the site are these little tiny lobe-finned fish that are about the size of a thumbnail. The biggest ones are about 15 feet long. So it's a fairly, it's fairly diverse fauna. Pretty much most everything above that thumbnail-sized animal is a carnivore. And pretty adept at that. And some of them, you would not want to swim in the Tiktaalik stream, let's put it that way.
I think it's lovely that you've got these huge specimens, but usually among the vertebrates there's more than one species, and they come in a lot of sizes. And I'm wondering if you're beginning to run across other species of smaller dimensions. That thing's huge.
Yeah. We do, and so the huge, from 4 feet to 9 feet is the range we currently have Tiktaalik. We have one bone that's about half that size from one individual, so maybe there's a 2 or 3 footer in there. But the question actually I thought you were going to ask, which is the relevant one, is we have different body sizes of these Tiktaaliks. How do we know whether it's one species or 2 species or a growth series or what have you? And we think it's a growth series right now. So. To answer your question, I think the smaller Tiktaaliks are likely to be found because we think we have a growth series, but that as you get to smaller size, you get to forms that have less fully formed bones. And because the amount of bony tissue is associated with how big these animals are as they grow, they add bone mass, we think. And so then if you believe that idea, then if you go to smaller creatures, they'll be more weakly ossified with less fully formed bones, and we may not be able to find them as easily.
You mentioned that you had found partial pelvis. Could you maybe say a little something about that pelvis?
It's got an ilium and an acetabulum. What to say about the pelvis? The pelvis has a very surprisingly socketed acetabulum, deep socket, which I would not have predicted. Very deep socket. Okay.
Thank you, Neil, for those great answers to those questions. Thank you all.